Herein, we provide a thorough description of laser microdissection applied to achieve a compared RNA transcript profiling of silenced and unsilenced areas of the Drosophila wing disc. This approach requires, as biological material, manually dissected Drosophila Imaginal discs, as main equipment, a laser microdissector. We used the Leica LMD6000 and a real-time PCR machine. We used a Biorad IQ5. Small tools required are a concave plate, a small paintbrush, microdissection forceps, hanging drop slide, insect pins mounted on syringe needles, metal frame with PET membrane, small plastic tubes. This method involves the following steps. Step 1. To cross two suitable Drosophila transgenic lines in order to trigger localized and specific gene silencing in the progeny, making use of the GAL4 UAS system. Step 2. To treat the dissection frames. Step 3. To set up the laser microdissector. Step 4. To manually collect the wing imaginal discs from the larval progeny of the genetic cross. Step 5. To perform laser microdissection of specific areas of the GFP labeled imaginal discs. Steps